talked about negative selection in B cells. All right, and you can use this chart, but anybody in person or via Zoom can chime in and kind of tell me what's a similarity and or difference between B cell selection and T cell selection. Jada? Okay, so they both have negative selection as part of the process, right? And so what's negative selection? Right, they get tested with self-antigens, right? And so if you respond to that self-antigen, right, we get rid of it from the system, all right? What else? So then what's the main difference that we see up here that's going on right here? What did we call that? Abby? Oh. And positive selection is? I'm glad that you said that instead of just pointing, instead of expecting me to, right, perfect, right. Binding, no, but like, well, yes, I can, right, perfect, right, Abby's right, right. Remember, T cell receptors cannot bind free-floating antigen, and so they need to have it presented to them, and it's presented to them via MHC. So, right, you have to be able to recognize or bind to an MHC molecule, that's a positive interaction, in order to hopefully generate a response, right? So you have the positive selection where they bind to MHC first, good, check. We then present to them self-antigen, all right? And if they respond to that, that's bad. Negative selection, we get rid of them, all right? And so you can see down here, right, apoptosis. And then, right, you have some that could become active T cells in future infections, all right? So really briefly, we're going to talk about lymphatic tissue, all right? Lymphatic tissue, right, has lymph vessels, all right? So if you've ever seen a diagram in an A&P book and you'll see like the red and the blue for the veins and the arteries, and then you see all the green vessels, right? All those green vessels are lymphatic vessels, right? And they're really important for fluid transport, fat transport throughout your body, and it turns out, right, lymphocytes, lymphatic vessels, transportation of lymphatic cells throughout the body, all right? If you've known someone that has edema, right? Edema is fluid buildup, and that can happen for a number of reasons, right? But the kind of basic premise of what's happening there is your lymphatic vessels, for a multitude of reasons, can't keep up with the excess fluid that's collecting in body cavities, right? And so then you get that swelling that occurs, okay? And in this case, right, these lymphatic vessels will go to certain organs and will go to what, when you go to the doctor and they're feeling here, they're feeling under your armpit, what are they feeling for? Lymph nodes, right? They're feeling for lymph nodes. All right, and lymph nodes are kind of like, I consider them like a sieve or a filter, right? They're kind of filtering out whatever's in the lymph to figure out if there's something bad in there. In your lymph nodes, right, you are going to have T cells hanging out, B cells, macrophages may come for a little bit, dendritic cells may come in for a little bit. It's a good centralized area for your immune system to figure out what's going on, right? If you didn't have your lymph nodes, you would just have cells floating around and you would be hoping for a chance encounter, right? That is not conducive to life. This centralizes everything so that cells can be activated appropriately, okay? Again, this is important if you've ever studied the lymph system. If you haven't, that's fine too. It actually connects with your venous system in your subclavian vein, so kind of up here, right? So it dumps all of its contents back into your circulatory system. It immediately enters the heart, where it can then be distributed throughout the body, right? And so not only is it important for fighting infections, it's also important for recycling fluids in the body. All right, so here you see a similar composition to blood plasma, and if you know anything about blood plasma, the majority of it is water, but like I said, this is actually an important way that fats are transported in your body, so it also has a high fat content, all right? And this is what I was talking about, all right? During your regular circulatory process, your blood pressure actually causes a small amount of fluid leak from your capillary bed, which is totally normal. Some of that fluid goes back into the capillaries, some of it starts to accumulate in the tissue, and the lymphatic system helps pull that excess fluid away from your tissue. Okay? If you want to know more about that, take AMP, when do we do that? AMP 2, right? Is when we do lymphatic system. So here you can just see a diagram of the lymphatic system, right? It does kind of mirror or follow your circulatory system, right? Kind of a similar setup. Here you have a lymph node, right? And in the lymph node, you can see these little nodules, right? So the lymph node is kind of divided into these little round, small segments. And in these round segments, right, this is where you're going to have T cells and B cells hanging out, waiting for macrophages or dendritic cells to bring them something, all right? And so when you're sick and they feel your lymph nodes, right, they can tell you're sick, or they can do a really quick assessment that you're sick when your lymph nodes are swollen. Because if, right, your macrophage and dendritic cells bring foreign antigen that your body responds to, you immediately start to have proliferation of those T cells and those B cells, right? And the lymph nodes start to expand or become swollen, all right? And then again, those cells can enter circulation by jumping in the lymph, right, back in these lymph vessels, going to the heart, where they're then dumped into the circulatory system, and they can be essentially rocketed to or shot to their target tissue. Okay? So again, we won't spend a ton of time on these, but you do have primary lymph organs and secondary lymph organs. So your primary lymph organs are going to be your red bone marrow and your thymus. We kind of have mentioned it, or we have mentioned it a time or two. Your red bone marrow, right, is where those hematopoietic stem cells can differentiate into something like a T cell or a B cell. In the case of B cells, the red bone marrow is where they go through their maturation process. In the case of T cells, right, they leave the bone marrow and go to the thymus where they finish maturing. So that's why we call those our primary lymph organs. Your secondary lymph organs, right, you have your lymph nodes, which we just mentioned. You have your spleen, which is really important for filtering or checking if there's pathogens in your blood. So if you've known anyone that has a splenectomy, right, where they have their spleen removed, they are actually more prone to blood infections, right? And so they have to keep a closer eye on that than, say, someone like all of you, I think, maybe have your spleen. I don't know. I didn't do a questionnaire, right? I'm assuming. If you don't have your spleen, please let me know and please tell me the story, right? Because it's an interesting surgery, right? Tonsils, right? How many of you have had your tonsils removed? Or at least some of them, you have multiple tonsils, right? You know, sometimes they just become so inflamed that it becomes an issue for airway, for speech, and all that stuff, so they can remove them. But your tonsils are a type of lymph tissue, right, where antigens can be sampled. And then mucosa-associated lymphoid tissue called MALT. Basically, within the colon and the small intestine, you have little special pockets of lymph tissue that are sampling the contents of your digestive tract. You think about it, right? Kind of Blair's article today was talking about bacteria in your colon. You have a lot of it. Bacteria in your small intestine, specifically your distal small intestine, you have a lot of it. And so it makes sense to have little pockets of tissue in there that are sampling those contents, because you don't want that to get out of control. In case you are interested, in the fall of 2021, right, I make you look at all of these things under a microscope. Huh? Jenna? You're considering it. I can see you looking at it. When do you graduate?
It'll be the spring.
Well played, Jenna. Well played. All right. Yes, ma'am. Well, okay. So for splenectomy, typically what happens there is that's some type of like traumatic injury that the spleen has to be removed because of something like internal bleeding or a lot of damage to that area, right? I'm not saying that I guess it hasn't happened from an infected spleen. If that does happen, that would be incredibly rare. Like I haven't heard of that. I'm not saying that never happens, right? For your tonsils, and this would actually be kind of a little bit, in my opinion, related to Jada's question, which I still haven't looked into, right? Why she got strep throat so much. Some people's tonsils, for whatever reason, are just overactive with immune cell proliferation. And, you know, some people might even argue that tonsils are kind of vestigial lymph nodes that maybe don't serve the same purpose that they used to. And so when that inflammation occurs, and it can't be controlled, then we remove them. Some doctors now, instead of surgery, will try to hit you with a heavy dose of antibiotics to get it under control. But one of the things I hope you can appreciate from a gut microbiome perspective is, although maybe that gets your tonsils under control, pumping yourself full of antibiotics, maybe it takes care of one thing, but then it can lead to some other issues. And so, you know, Atlantis, I would have to look into, you know, is it some type of, you know, bacteria that causes that overreaction? But certainly you can have your tonsils removed, right, and be relatively normal, right? Did you have your tonsils removed? No. See, I could tell. But I'll check into that, you know, for you, what specifically, you know, makes that happen. And I'll say this too, and the structure of tonsils is kind of strange. It's more of like a pit, and things can get stuck down in there. And so I'm sure that contributes to some of the inflammation, which is not necessarily what you want. But I'll see if I can look into that for you. That's a good question. All right, we're going to skip this video right now because it doesn't play well. Antigen processing and presentation, right? Well, here, I'll show you some diagrams for that stuff in just a second. And I think that was part of your mastering bio experience to look into some of that. So we'll talk about that here in just a little bit, hopefully. One of the things I wanted to talk about, though, that's really important, right? And antigen presentation is incredibly important, so please don't think that I'm dismissing that. But it's this idea of clonal selection. And so we've talked about specific responses to specific pathogens by your immune system. We've hinted at this idea of immunologic memory, right? So when you see an infection once, you develop memory cells. So if you've seen that same infection again, you hopefully will be able to respond much more quickly. And so that's what clonal selection is, right? It's taking a naive B cell or T cell, exposing it to foreign antigen, right? Getting it to proliferate and divide and respond to that pathogen, and or make memory cells for a second encounter with that pathogen. What I kind of focus on here on these diagrams is B cells, but please note that T cells go through a very similar process. So if I were to ask a question on an exam about clonal expansion, I would want you, right, to be sure that you point it out to me, that you realize the process is occurring for both T cells and B cells. It's very, very similar, but if I'd asked something about clonal expansion, and I'll just use her as an example because I know she would never do this to me, and Jennifer Buckner just talked about B cells, right? She would get probably a lot of credit, but she wouldn't get the full credit that she was seeking, right? So be sure that you remember clonal expansion applies to T cells and B cells. Regardless of what cell you're starting with, a T cell or B cell, you start off as an immature lymphocyte, right? And I'll tell you specifics of what that means in just a second. You then become a naive lymphocyte. You become an activated lymphocyte. And then you become an effector lymphocyte and or a memory lymphocyte, all right? So there's basically 4 kind of basic categories that we put these cells in. Yes, ma'am. The lifespan for a lymphocyte when it's activated, I'd have to go back and double check. I believe it's a few days. So they're not around incredibly long, except you're always generating new ones for an infection and kind of dumping them back into circulation. But I'll double check. Hey, Ajax, can you do a quick lymphocyte lifespan search for me? All right. So here's clonal expansion in a B cell, but please remember, T cells are also going through a very similar process. So if we go all the way back up, we see here that we start with hematopoietic stem cells. All right, that hematopoietic stem cell, right, receives a signal and it starts to differentiate into a lymphoid, right, in this case, a B cell. And the first kind of step here is an immature B cell. If you look at this diagram, you'll see that an immature B cell is a B cell, but it is lacking expression of the B cell receptor, right? It's immature, so it doesn't have those markers on its surface that we associate with a functional B cell, all right? It eventually does get those markers on its surface, and it becomes a naive B cell. So it now has a B cell receptor on its surface. And this B cell receptor, if you remember, we talked about that RAG enzyme that was rearranging antibodies, right, to give us these different combinations. And this B cell receptor, right, is presumably looking for some antigen, right, that may be present in the system. And I like this diagram because it shows you B cell W, X, Y, and Z, not overly creative in their name, but it does indicate, right, that each one of those letters is saying, hey, this B cell receptor is for a specific antigen. And so in this case, antigen X is only going to be able to stimulate the B cell with the B cell receptor X, all right? These other B cell W, Y, and Z, right, aren't responding to antigen X. It does not mean that they will never do anything. It just means they need that specific antigen present, all right? So your naive B cell, right, same process applies to T cells, all right, is interacting with a specific antigen, all right? This then allows us to be activated. Now, recognition of an antigen is not enough for activation, all right? Cells need help, right? Whether you're a T cell or a B cell, this activation step is always going to require the help of another cell, all right? So in the case of B cells, you can see here, this help is coming from a T helper cell. Good for it, right? It gets to do its job, right? So T helper cells are providing a stimulatory signal to a B cell that has recognized a foreign antigen. All right? So I've recognized my antigen. I've gotten, in this case, T cell help. I can now actually proliferate, and differentiate into plasma cells that are going to produce antibodies to fight the infection, and then memory cells, all right? So you're doing two things, right? You're responding to the current infection via the plasma cells, and, right, you are anticipating a future infection, and you have these memory cells that are now hanging out, right, and are ready to go. Ajax, upon activation, how long does a lymphocyte live?
So, naive T cells can live for up to 9 years.
Woo, naive T cells, 9 years. I'm talking about activation. Give me activation. Yeah, I want activation.
T cells are active for 50 years after you get smallpox vaccination.
That's a memory cell. Ajax, interrupt me in a minute when you get it. I believe in you. All right? All right. So I want to point out really just a couple small differences then between T cells and B cells, right? So this is great for B cells. What's different for T cells? Well, remember, T cells don't bind free-floating antigen. And so a naive T cell will have a T cell receptor on its surface, but it will have to have something bring the antigen to it, right, via that MHC complex. But whatever is bringing that antigen, right, presumably will be able to provide those stimulatory signals. And so if we're talking about T cells, more than likely what is helping a T cell during clonal expansion is going to be a macrophage or dendritic cell, right? A macrophage or dendritic cell can release those stimulatory signals that help lead to T cell activation. Upon T cell activation, you have proliferation. So you'll have, in this case, we would just call them effector T cells that are engaging with the pathogen. And then you will have your memory T cells that, again, can remain around for a long period of time, just like my good friend Alyssa Jackson in the back mentioned. All right? So again, it's a really similar process. It's just, hey, remember, B cells can bind free-floating antigen. B cells are more than likely getting help for activation from a T helper cell. T cells, right, can't bind free-floating antigen, so something has to present that antigen to it. But, right, whatever's presenting that antigen, presumably a macrophage or dendritic cell, is providing the help that it needs for activation, boom, proliferation, yada, yada, yada. Alyssa? That helps activate a B cell? Yes. Yes. Well, I say that there are some rare, interesting exceptions, but for the sake of me right now here, yes. Okay? Well, in this case, because we're going to say this is happening in a lymph node, and so what should be presenting an antigen in a lymph node is typically going to be a macrophage or dendritic cell. All right. Again, there are always some interesting exceptions. For me, that's what I want from you. All right, Caitlin, and then I'll circle back. Did you have a question? Caitlin, that would be probably an evolutionary biology question, which I'll ask you to send to Dr. Freak. I don't know, right? That's part of the design of the system that they can't. I would say from a pathological perspective, it allows a tighter system of checks and balances, the fact that something has to present it to it. If it can bind free floating, then there's a greater chance that it could react to something that it shouldn't, or you couldn't regulate it as well, right? But as to exactly, and there might be some interesting, animal that's not as developed as us that has, you know what I mean? So I don't know where that kind of divergence went. That's a good, interesting question, but I'm not sure. I have no idea. All right, Jennifer Buckner.
So lymphocytes come from a hematopoietic cell, but can they divide themselves? Like they just have to come from
Okay, so yes, I think if I'm understanding your question correctly, so they come from this hematopoietic stem cell, right? And remember, you have the myeloid lineage and you have the lymphoid lineage. So in this case, we're looking at the lymphoid lineage. There's a series of signals that cause them to become a T cell or a B cell, right? Once they become a T cell or a B cell, I mean, they can divide if they receive this stimulating event. Is that what you're?
So that's how they keep the memory cells going?
Yes. And so what would happen the next time you had an infection is this memory cell would become activated and it would basically jump right to this step here. So this memory cell would lead to the formation of plasma cells to fight that infection, but then it would also lead to the formation of new memory cells. So upon activation, you're always kind of starting this proliferation and differentiation event again, assuring that A, you fight the infection, and B, you make sure you have memory cells for the next time you see that infection. Does that help? Perfect. Abby, are you good? Michael? T cells do also form memory cells, yes. So the basic principle of vaccination, right, is relying on the formation of these memory T cells and these memory B cells. All right. And some of the vaccines that they're looking at actually do a better job of getting memory cells formed from either one. Like a good vaccine would get memory T cells and memory B cells to form. Yes, ma'am. Could be different, right? So the T cells, you could have T helper cells that go through clonal selection. You could have cytotoxic T cells that go through clonal selection. But if we're talking about B cell clonal selection, it's going to be a T helper cell that aids in this process. A cytotoxic T cell is typically not going to aid in the clonal selection of B cells. Does that make sense? You promise? Okay. If it doesn't, I expect an email from you. Deal? Who was? Oh, Zach. So Zach, theoretically, as long as that antigen is present and is able to stimulate, then you'd keep on proliferating and responding to it, right? Good question. Anybody else? All right, let's talk about antigen presentation here as quickly and responsibly, I guess, as we can. Yeah.
It takes like 5 to 6 days to activate a T cell, and it lives for about 6 months.
Oh, 6 weeks. I was way wrong. There we go. 6 weeks. All right. So it's around for a while. All right. So reset. Okay. So let's talk about antigen presentation. So B cells, right, are interesting because although they can't phagocytize, right, they are actually really important for antigen presentation. So what you see here is this fun Rubik's Cube binds to a B cell receptor. Upon binding of that Rubik's Cube, aka that pathogen which has antigens with it, it is taken in via endocytosis, and it's broken up, and then it's presented on the surface via MHC class 2. This is an important thing to realize. MHC class 2 presents antigens that were phagocytized, or in this case, right, B cells are a little different, but were taken in from the external environment. All right? And MHC2 is going to present specifically to the T cell receptor on a T helper cell, right? And here you can see upon recognition, you have that cytokine delivery that leads to the activation and then proliferation and expansion of your, in this case, B cell. All right. So when a cell eats something or a cell brings in something from an external environment, right, in this case by the B cell receptor, it's broken up and it's presented on MHC2, which then presents to the T cell receptor on a helper T cell. Got that, Hannah? True? Michaela? You got it? All right. What?
So on macrophages and dendritic cells, when they're presented, that's MHC1.
Macrophage and dendritic cells and B cells are pretty unique. And we'll just skip here for a second. MHC1 is present on pretty much every cell type that exists in your body, all right? MHC1, the way I look at it, is it's basically a window into what is going on the inside of a cell, all right? MHC2 is only on cells that are able to phagocytize, or in the case of a B cell, right, bring in its B cell receptor via endocytosis. That is why dendritic cells and macrophages primarily are such important cells for the immune system, because they have MHC1 like everything else, but then, right, they possess MHC2, which is unique to just the regular epithelial cells in your body or the cells of your connective tissue, all right? MHC1, all right, presents endogenous antigen. So it primarily, right, is going to be presenting one of two things. More than likely a viral infection, right, because viruses infect cells, right, and they hang out in the cytoplasm, they do all their stuff there, so that gets presented. But what's interesting about MHC1 is that your cells are constantly remodeling and are changing and breaking down protein to build something new. And so MHC1, right, oftentimes will contain a lot of self-antigen also, right? So then it really is up to the immune system to say, okay, is the MHC1 on Sarah Martin cells, is it showing me a pathogenic antigen, more than likely from a virus, or is it just showing me a piece of broken down protein because she's remodeling her cell cytoskeleton, which is a total normal function, all right? And MHC1 is going to activate those CD8 T cells, okay? So CD8 T cell, right, the T cell receptor on a cytotoxic T cell is going to recognize MHC1 and then determine, hey, is it infected or is that just a normal antigen, so I'm going to leave it alone. Right? And this is just showing, again, MHC2 to kind of give you a side-by-side comparison there. So does that kind of basic premise of antigen presentation make sense? So MHC1 is presenting to CD8 or cytotoxic T cells, right? Present on pretty much every cell of your body. Again, interesting rare exceptions that we're not going to really get into here. MHC2 present only on cells that are capable of phagocytizing, all right? And then, you know, B cell, you might not call that traditional phagocytosis, but in this case, right, we do consider it a type of phagocytosis, all right? And so that's why antigen presenting cells have a unique function, because they can interact and activate T helper cells, which can activate the innate immune system and can also activate other cells of the adaptive immune system. All right. Those of you that are super interested in this, right? Well, I don't know if Dr. Chrisman will be offering immunology before you graduate, but if he does, take it, right? And you could spend a whole unit on antigen presentation if you want to. All right. So again, this is everything that I just told you here, all right, about T cells and B cells. We've talked about this lymphoid tissue. This again is kind of showing the uniqueness and the importance of dendritic cells. This will be where we end. I think we got through, let's see, most of it. Yeah, the last little bit is a little more an antigen presentation, so you can look at that on your own. I don't think there's anything there that's incredibly complex. I think we've kind of set the table for it. It's only a couple slides, so just please take a look at them. I'm not going to give you a 20-point essay question on the last 3 slides on this presentation that we didn't get to. All right, don't worry about that. But I do love this because it shows the importance of dendritic cells. Remember, macrophages can do this, right? But they're a lot of times concerned with just, I'm going to eat, I'm going to fight this infection, and that's my job. Dendritic cells, right, they're more, you know, like a foodie with a cheese plate, right? They like to nibble on a little bit of this, a little bit of that. Yeah, you know what I'm talking about. Yeah, you're a cheese plate foodie, right? And so they're just kind of sampling, but they're really great at sampling, and it's really important because, again, their MHC2 and MHC1 allows stimulation at the same time of both CD4 and CD8 T cells, all right? So dendritic cells can kind of, right, in one meeting, amp up the entire immune system. Man, we love dendritic cells. Macrophages can do this too, but it's not their primary job, right? The dendritic cell is the professional APC, right? It's the cream of the crop. It can activate almost the entire immune system with one interaction. All right? Cool. So I'm a couple minutes over. I'm going to let you guys go. Please look at these last couple slides. If there are any questions, comments, concerns about them, shoot me a quick email. I don't think it should be anything crazy because, again, I did assign adaptive immunity on the mastering thing, and I think a lot of this was hit in that. But please send me questions. Please remember I will open that exam at 7:45 on Friday, a.m.